Good morning, everyone. On behalf of Deshbandhu College, I extend a very warm welcome to the respected chairpersons, distinguished speakers, and the audience to the third and final day of International Conference on Integrative Chemical Science for Health and Environment. Now, before we begin with the invited talk for today's session, I would first like to introduce the chairpersons for today's session. We have Dr. Shailendra Narayan Sharma and Dr. Shubhra Chaturvedi with us as chairpersons for this 8th session of the conference. Dr. Sharma is presently working as a senior principal scientist in CSIR National Physical Laboratory, New Delhi. Dr. Sharma did his PhD from Indian Association of Cultivation of Science, Kolkata. He subsequently did his postdoctoral research from University of Barcelona in 1997. His area of research interest includes synthesizing and characterizing various types of semiconductor nanoparticles. Dr. Sharma is also working on polymers, organic, inorganic hybrid materials, and nanocomposites. Dr. Sharma has many publications in journals of national and international repute and has supervised many PhD students. Our next chairperson is Dr. Shubhra Chaturvedi. Dr. Shubhra is presently working as Scientist E at Institute of Nuclear Medicine and Allied Science in Mars, DRDO, Delhi. Dr. Shubhra did her master's in chemistry from IIT Delhi, and subsequently she did her PhD from Banaras Hindu University and Institute of Nuclear Medicine and Allied Science in 2013. Her research work focuses on development of novel radiopharmaceuticals for application in oncology and neuroimaging. Her core competence is in computer-based drug design method, organic synthesis, in vitro method based on cytotoxicity assays, and nuclear imaging techniques. Dr. Shubhra is member of many scientific societies, such as Society of Nuclear Medicine, Indian Chemical Society, Indian Society of Nanomedicine, and American Chemical Society. Dr. Shubhra has supervised many PhD and master students, and she has many research publications to her credit. Welcome to both of you, sir and ma'am. And with your permission, can we start the session now? Over to you. Thank you so much.
Good morning to all. We will start the session today with Professor Virendra S. Parmar's lecture, invited talk. And he is basically a faculty in nanoscience program at the CUNY Graduate Center in Chemistry at Medgar Evers College and City College, the City University of New York, USA. And before he starts his lecture, I would like to give his brief introduction. Professor Virendra Parmar did BSc Honors, MSc and PhD from the University of Delhi and has worked for nearly 10 years as a postdoctoral visiting scientist at Cornell University, Harvard University, Massachusetts Institute of Technology, University of Massachusetts Lowell, NYU, Poly, University of Basel, and Imperial College London. He is currently a faculty in the nanoscience program of the CUNY Graduate Center and in chemistry at City College and Medgar Evers College. Earlier, he had served the University of Delhi for 44 years as professor and head of the Department of Chemistry and as chairman of the Board of Research Studies and provost of Guer Hall. In recognition of his excellent services of 44 years in teaching and research, the University of Delhi conferred him with a Distinguished Service Award for the retired teachers during its centenary celebrations in March 2023. He was appointed full tenure professor of chemistry and first head of the Department of Nanoscience at the University of North Carolina. He has been an awardee of medals for excellence in research from the Chemical Research Society of India and of the Indian Society of Chemists and Biologists, and a recipient of the Academic Staff Award from the Experts Second Consortium of the European Union. He was awarded the Dean's Research Excellence Award of the School of Science, Health and Technology, MEC, CUNY, New York, USA for the years 2019 and 2020. Professor Parmar's research interests include biotechnology, green sustainable chemistry, nanotechnology, organic synthesis, medicinal chemistry, and the chemistry of natural products. He has mentored 85 PhD and postdoctoral scientists and has published more than 500 research papers and obtained 11 patents as well. So over to Professor Parmar for his illuminating talk. Please, sir.
Good morning, everyone. And thank you very much, Dr. Pratibha Kumari, for inviting me to have participated in this wonderful meeting. I'm sorry I could not attend the earlier sessions, but the topic of the conference is very pertinent in today's scenario. And I immediately accepted to be part of the conference and share my thoughts on a very hot topic today, deals with both environment and medicinal chemistry and human health. That is antimicrobial resistance, AMR, popularly called. And thank you, Mr. Chairperson, for your very kind introduction and your words of praise and appreciation for me. I thank you very much for that. Now, I know that we are already a little late in starting this session, so I do not know how much time I may have. So I will very quickly go through the slides and share with you some important points dealing with the topic, because as the topic of the conference, or the title of the conference is Environment, the Chemistry in Environment and Health, basically. So this is very much, the AMR is of great concern to human health today. So I will not talk more about myself, because already a lot has been said in the introduction. So can I go to next slide? Because I cannot control slides from here. Yes. So this is a graduate center, which is the CUNY is a system like University of Delhi. It is made up of 25 colleges, like University of Delhi, which is made up of 93 colleges today. So, and the graduate center where the master's program is there and PhD program is there. In the colleges at CUNY, we have undergraduate teaching. Next slide, please. I always dedicate my talks to this great man. I do not know how many in the audience would know about him. But he is the, in a way, father of the Department of Chemistry at University of Delhi. And I had good fortune of having been taught by him in master's and then very early years of my PhD research. So I call him my guru, which is, now this is just, next slide, please. That's right. Please, next slide. Yeah, I just want to tell you the importance of natural products in human health and also in medicinal chemistry. So just next slide. Yeah, this is the most complex, perhaps, organic molecule ever to be isolated from a natural source, a palitroxin, made up of 115 carbon atoms contiguously attached to each other with a lot of stereocenters. And it can have more than 1,000 possible isomers, out of which only one exists in nature. So it was reported in 1989 in C&EN, and it was called Mount Everest of chemistry. Next slide will tell you why it was called Mount Everest. Oh, sorry. So the structure is not being exhibited, so we can jump the slide. Next slide, please. So as far as our areas of research are concerned, I was trained as a natural product chemist, the area of research which we continued even till today. A lot of work and publications have been made in this area in collaboration with several research teams in Europe and USA and some chemical industries also. Next slide. Please move on this slide. It will show the different slide, the areas, yes. So then we moved on to synthetic heterocyclic chemistry, then biocatalysis. Perhaps we obtained a niche in this area as we started this area in India. Maybe we were the first groups in mid-80s to start this area, which is of direct importance today in industry in achieving the goals of green synthesis. So then we moved on to nanomaterials, and these are the funding agencies which funded our research both in India, CSIR, DST, DBT, foreign agencies like Danida in Denmark, DFG in Germany, and several others. So we were able to publish about 516 research papers in collaboration with 26 research groups in 15 countries. Next slide, please. So if this is the world health today, scenario, if you see the mortalities due to different areas of diseases, the infections is a huge chunk of mortality. About 13.3 or 14 million people die per year worldwide because of infections. Infections caused by bacteria, fungi, molds, and many other microorganisms. So this is a very serious area to dwell upon. Perhaps the other area which overtakes the deaths due to diseases is cardiovascular. So we have done research in both these areas, but I will talk on infectious diseases today and the seriousness of AMR. Next slide, please. Now, this is antimicrobial resistance, AMR, which is a global concern today, not only in India, but more so in India because of our population and the carelessness of people towards using antibacterials, even in cases where they are not required, and overusing them, and easy availability of them over the counter in pharmacies. Unlike in the West, in the US or Europe, where it is very strictly monitored. So we will talk on that. Next slide, please. One thing I can say, the slides are with the college and the host. Anyone who is interested can look at in more detail after the presentation. So I will quickly go through because of the shortage of time. Now, this is the milestones in my antimicrobial chemotherapy. We all know that the field started with Alexander Fleming's discovery of penicillins during World War II. Because of the great need, more than a few million soldiers or the fighters died both sides from the Allied forces as well as from Hitler's army, because of the infections which were caused due to weather conditions and due to the other environment reasons. But then the penicillins or beta-lactams, as we all know, then other types of antibacterials were discovered. But there was a huge gap in drug discovery of antibacterials between 1960 and 2000, because people thought at that time that we have a big arsenal of antibiotics of different kinds to kill all bacteria, which turned out to be wrong in 80s, 70s because of the mutations in bacteria. So the available antibacterials were not effective in killing the bacteria. So there was a need to have more new and novel antibacterials, but there was nothing in the battery, in the pipeline, in the industry and academia. So there was an innovation gap in this research area for 40 years, which I think was a disaster. So I think, and this was beautifully written by my friend, Christoph Walsh at Harvard Medical School, published in Popular Science, scientific magazine in 2006. So this is, I think, a very awakening for all of us that we should never leave innovation and research in any field of study, and there should not be a gap. And this was a huge gap of 40 years. Next slide, please. So next slide. So I talk of antimicrobial resistance, and it is, I like to this title, Glutter to Gut. So how from the glutter or the gutter, the bacteria have been reaching the gut, human gut, and causing all these diseases. And many of these bacteria have become resistant to the available antimicrobials. So how the antimicrobial resistant microbes journey from environment to humans? Antimicrobial resistance genes can be transmitted from environmental sources to the human gut and vice versa. So it is not one-way traffic. So human people who are affected by these serious infections, then the bacteria, untreated bacteria, goes into the gutter and travels back into normal people, healthy people, and affects them. So that is very serious thing, and we need to find antibacterials which are very effective. Next slide, please. Next slide. So I think this is a very, no, last slide, previous slide, please. Yeah, this, I want to read this to you because this from here, real cause is there more so in our country, in India. So this was said, the discoverer of the first antibiotic, penicillin, by Sir Alexander Fleming, a British scientist. And he got Nobel Prize within his 2 years of his discovery because of the immense importance and application to people, at least soldiers at that time who were affected and died during World War II. So in his lecture, Nobel lecture in 1945, said the time may come when penicillin can be bought by anyone in the shops. Then there is a danger that the ignorant man may easily underdose himself and by exposing his microbes to non-lethal quantities of the drug, make them resistant. Today in 2021, I think I should talk of 23, the same story. Doctors warn us against the misuse of antibiotics and how antibiotic resistance is a worldwide threat. What is antibiotic misuse? Misuse of antibiotics refers to consuming antibiotics without medical advice. In our country, more so. So even if we have a little bad throat or bad cough, we start taking streptomycin, we start taking erythromycin because they're easily available. Reusing old prescriptions because they're outdated, they will not be effective. So not completing the prescribed course or shortening the treatment duration, taking less than the prescribed dose, or failing to comply with the right frequency. So all these causes are all these reasons are the causes for AMR, which is very serious threat to human health today. Next slide, please. So what is a superbug? Superbug is one which is untreatable and which is present all around us. So this was a real happening in Vellore in Tamil Nadu. There have been reports of other superbugs arising in India. Just one case we are discussing here. Last year, in 2021, we're talking of scientists at a hospital, CMC, Christian Medical College, very famous hospital, in Vellore reported a highly virulent form of pneumonia. It was resistant to carbapenem antibiotics, which are given in such infections, which are reserved for fighting multi-drug resistant bacteria. More than half of the patients who reported this strain succumbed the infection, an unusually high mortality rate. So this is very, very serious. Next slide, please. So this is the founder of NDM1, another very celebrated doctor, scientist, Dr. Lakshminarayanan, who writes this. Even in the US, there have been cases of this AMR, where the tight control of antibacterials supply or to be bought is there, very tight. You cannot buy even one tablet without doctor's prescription. Next slide, please. I think I leave this. Next slide. Genes can be transmitted from environmental sources to the human gut and vice versa. From sore throats to fevers and life-threatening infections, most people have periodically used antibiotics, even without doctor's advice. Recent reports show that the global COVID-19 pandemic has increased the use of antibiotics. People take them because people who are not familiar with the COVID infection when the pandemic was there and when medications, vaccine was not developed and available, people in the rural areas in particular, and even the cities, they thought it is an infection. So just took antibiotic and took them. So they were not effective because they were not meant. COVID was a viral infection, not a bacterial. So the antibacterials were taken because over the counter they are available easily all over India. And so they were passed on to the environment from feces or urine or some other excreta. And then they traveled back into the human beings. So this was a cycle, deadly cycle. Next slide, please. So I will again skip this, I think, because of lack of time. again, but there's beautiful readings here. So what are antibacterial resistance genes? Globally, health problems due to antimicrobial resistance have been rising. One of the major reasons is the mobility of ARGs, which are antimicrobial resistant genes. So the resistance genes can be transferred from one generation of microbes to the next through reproduction and can also be transferred by horizontal gene transfer. So they both are either the genetic transfer or from one species of microorganism to the other. So it is a very fast transmission among bacteria, and all become mutated and resistant to the antimicrobials available. This implies that regardless of the origin, antimicrobial resistant genes can be transmitted from environmental sources to the human gut and vice versa. Next one, please. So they can come from wastewater, which contain plenty of resistant genes, from human feces, farms, households, and hospitals. Next one, please. From food, to increase animal productivity, right? Because to produce more meat, which is chicken or other animals like goat or beef, antimicrobials are often applied on farms to keep these livestock alive for longer time. And this can then, the antibacterials can then pass on to the environment. Through the wastewater treatment, they can pass on to human beings. So this is seen that then this can be transmitted to edible plants such as tomato, lettuce, and other many crops. And from air, inhalable antimicrobial resistance genes pose an increasingly growing silent health threat through the air. Next slide, please. From healthcare facilities, because if the biological waste generated in hospitals and clinics and dispensaries is not properly disposed, that's a very high risk source to spread high antimicrobial resistant genes. And travel, of course, air travel or train travels, people carrying diseases can spread them to other people traveling along with them. Next slide, please. So this was a very nice term given, One Health approach. It is evident that antimicrobial resistant microbes surround us, and it is normal to feel concerned. But what is even more important today to recognize are the tiny steps we all can still adopt. This includes ensuring patients are treated with the appropriate antibiotic in the right dose, and really the ones who need them. Next one, please. So the conversation is very important between the doctors and the patients and the hospital staff. Next one. With resistance to first and second generation antibiotics, doctors forced to use reserve drugs. So because of this ignorance, or even, I think, the improper use of antibiotics among the people, the educated people, so the first and second generation antibiotics available in the market, even to them, the microbes have become resistant. So then doctors give reserve drugs, means those which are rarely used or very rarely used because of the need to treat the patients. So therefore, even they become resistant. So this is, I think, very serious cycle of events. Next one. So this is a real crisis, and crisis is above pandemic. Right. So this is, I think, a very, very nicely put slide that more than 2 million people in India alone die because of AMR. Next one. Poor antibiotic stewardship, blamed as India found to be superbugs' birthplace. I think this is very serious thing for in our country, in India, to think about, because of our large population and because of easy availability of antibiotics over the counter. Next one. So this has been done very thoroughly in many commonly found bacteria, Staphylococcus aureus or E. coli or others. Next one. Now, this is, I think, the last general slide which I wanted to share with you before discussing our own work in this area. ATLAS is a very popular synonym which stands for Antimicrobial Testing Leadership and Surveillance. And this is a global, including India, fully searchable database developed by Pfizer, a leading pharma company which developed the first COVID vaccine for antibacterial surveillance, which is useful for the health practitioners to understand local resistant patterns of various bacteria and the antibiotics that can work on these bacteria. We also have ongoing educational programs for clinicians, nurses, and pharmacists on implementing antimicrobial stewardship in hospitals. So this is a very important program which has been adopted by the government of India through ICMR. So I think you should read more on that and make yourself aware and your communities aware that this is a very serious threat to human health now and in years to come more so, unless we are very careful to change our habits of taking drugs unnecessarily over the counter, even in small infections which may not need the antibiotic, we still go and buy and consume it. Next slide. So the conclusion is need of the hour is proper training of healthcare workers regarding prescription of antibiotics and awareness regarding stopping the use of unnecessary antibiotics among the public. We need laws to prevent over-the-counter sale of antibiotics. I think that is strongly desirable, and the Health Ministry of the Government of India, as soon as possible or sooner than later, should adopt this, that the antibiotics should not be sold over the counter and strictly ban the sale of antibiotics without prescription from a trained doctor. All these steps will help to curb the spread of antibiotic resistance. Next slide, please. So I will talk of two areas of research, as I have said in my title, what we did in kind of dealing with the serious infections which were not treatable by available antifungals or antibacterials. So the first is a fungal infections disease, which is aspergillosis. And this work was done since 2005 in collaboration with a research team at CSR lab on the campus of the University of Delhi, IGIB. Next slide, please. This is aspergillosis. It's a serious lung disease, which, if left untreated for a while, can cause death. It is not very prevalent, but still it is very serious if it happens, particularly among immunocompromised patients and older populations in the hospitals. So this is caused by fungi, two species of fungi belonging to Candida and Aspergillus families. Next slide, please. So this is the way Aspergillus infection happens. Aspergillus fumigatus is the major culprit, major species that causes this. It is superficial allergic invasion, goes through the respiratory canal to the lungs, and they are affected. So this is initially confused or mistaken for tuberculosis. So the usual drugs for tuberculosis are given, and by the time the patient does not respond to them, it is too late, and they die of aspergillosis. So early diagnosis and treatment is of absolute necessity in this area. Next slide, please. The only drug which was available till today was this very complicated, complex structure called amphotericin B or nystatin. So this is effective, but it is highly toxic. It may be killing the fungal or fungus causing the aspergillosis, but it causes other toxicities to normal healthy cells also. Or B, it is highly difficult to obtain this synthetically, So only source is natural, which is limited. So both these factors, the limited availability and also that it is highly toxic drug, it seriously needs other alternatives for aspergillosis, a fungal infection. Next slide, please. So these are limitations of current therapy using this drug, which I just shared with you. So this necessitates to identify novel compounds, molecules for development of new antimycotic formulations against aspergillosis. Next one, please. So we saw that azoles are very commonly used antifungal agents. The triazoles tend to have fewer side effects out of azoles, like imidazole. So better absorption, better drug distribution in body tissues, and fewer drug interactions. So the triazole-based antifungals are very popular. I will show a few on the next slide. Next one. Yeah, this is the antifungal azoles. Voriconazole, very famous. Fluconazole is the most famous and popular triazole-based antifungal agent. Next one, please. So next one, I will concentrate on fluconazole. Next one, please. Yeah. This is the antifungal agents in the market, and we will discuss fluconazole because it is the most effective. It contains 2 triazole rings in the same molecule, and it's prochiral. Means if you make a change in one of the substituents, you can make the compound chiral. So that is the property or the structural feature of fluconazole, which we took advantage of to make it better for treatment of aspergillosis, using our knowledge of biocatalysis and nanotechnology, so-called bio-nanotechnology. Next one, please. Next one. So it was very effective in treating the, previous slide, please. It was very effective in treating the superficial infections on the skin or even in the mouth. You can see here that you can treat the infections caused by different infections in the mouth, on the tongue, or on the lips, or on the jaw, and hands, feet. But in the lungs, interior of the body, it was not effective. So we wanted to change the structure of fluconazole so that we can make it effective against aspergillosis, which is a lung disease. Next one. Next slide. So you can see this slide of, I have just put the whole work on one slide, but this
excuse me, Professor Parma, you have 5 minutes left.
Okay, I will be very quick. So this is just one slide to show you the entire summary of the work, which was published in 2017 in collaboration with various teams in US, in UK, and France, and of course, CSIR team and the University of Delhi, where we developed this agent by manipulating the structure of the prochiral fluconazole and make it chiral derivative by a biocatalytic reaction using the lipase from Candida antarctica, and then encapsulating it in cyclodextrin. Next slide, please. And that became more effective. And this is the table which shows the whole scheme. And we can see this, that it became very effective, as the next table will show you. Next slide. You can see here the effectiveness of this new variant of the structure of fluconazole, which we obtained by biocatalysis and nanotizing it. So it was at least 100 times more active than polytoxin, which is toxic and in limited amounts. So this, I think, applying the knowledge of biocatalysis and nanotechnology, we could develop a nano medicine in a way which is much more effective in killing this resistant strains of the fungus. Next slide, please. So this is a concluding remark on this antifungal development. Next one. Next slide. Now I talk to you again, please, previous slide. This was the work done, previous slide. Yeah, this was, I think, most deadliest bacteria known to mankind is Clostridium botulinum, called botulism. And this work was done in collaboration with the leading team at University of Massachusetts in Dartmouth, near Boston. So we got screened our battery of natural products from my collection at University of Delhi. More than 300 compounds were tested, and we found one which was very active. And we published quite a few papers. Two are shown here, one in 2021 and other is 2018. Next slide. So as you can see here, the fatal dose is that just 150 nanograms of this bacteria can kill a man of 150 pounds or 75 kilograms, normal human weight. So this is the most deadliest bacteria known to, and there are no antidotes to this. And this can easily spread through environment. Next one. Next one. So I've just 3 slides more just to show you how this collaboration was started in the year 2009. And very intensive work was done by 2 PhD students, one in Delhi and one in the US. So we screen, next slide, please. So this is a biothreat also. Next one. So antidote against the most poisonous toxin. There's no successful antidote. Next one. Next one. Next one. I'm jumping through slides. I should have discussed them. So we subjected a battery of more than 300 compounds. Next slide will summarize that work. Next slide. Yes. More than 300 compounds were tested, and they all showed some kind of activity. So finally, 4 compounds were pinned down, which showed 70% inhibition of Clostridium botulinum. And finally, 1 compound turned out to be most active. It was collection number in our collection, MLN 4493. That is my list number. Next one. So this was a furocoumarin-based compound, which occurs in Soralia coralifolia, which is very commonly occurring plant in Rajasthan, Tamil Nadu, and many other arid regions of the country. Next one. So this is a plant from which these furocoumarins or soralanes are isolated. Next one. Soralia coralifolia. So this coumarin structure is shown on the left top. In the top right structure is soralane, which occurs in that plant, Soralia coralifolia, and which is very commonly used drug to treat psoriasis, skin infections. So we found that a variant of this, which having a 4-nitrophenyl ring at carbon 3 position of furocoumarin soralane, was most effective. We abbreviated this as NPP, the chemical name or the IUPAC name of the compound. Next slide, please. So you can see here the structure of this compound. And next slide, last slide, shows the binding. There are 7 kind of forms of botulinum, Clostridium botulism, called BONT-A to Z. The first one, BONT-A, is the most deadly. So our compound, NPP, you can see here, it has a right fit into the active site of the enzyme, the protein of the BONT-A. So it gets deactivated. And that is the mechanism of the action. So I think we were very happy that a natural product-based compound could be found as an antidote for this resistant bacteria, the most deadly bacteria known to mankind. Next slide, please. So I have to thank a large number of people who came and joined our research group at the University of Delhi, and they later went on during their PhD to various labs in Europe and the US, Canada, UK, and Japan also. So, and they did excellent work both in Delhi and those labs abroad in collaboration. So I thank all of them for their dedicated work. And I thank a lot of funding agencies, both in India and outside, in the US, in Denmark, in Germany, and my collaborators. I strongly appeal to all the people who are still active in research, and also the younger lot, the students who are doing PhD or about to start research careers, that please establish collaborations. Without collaboration, we cannot do anything. And then sustain collaborations. Finding a collaboration, initiating a collaboration is one thing, easy. But sustaining it over the years, I think it is most challenging task. For that, you need to develop mutual faith and confidence in each other. You have to give the due credit to your collaborators and his team in authorships on papers, authorship on patents, and other recognitions. So, and I think because we kind of followed all those scientific ethics and ethos of collaboration, our collaboration is still running in many places for the last 30 years, 25 years, and still publications are coming in under collaboration, joint authorships. So new collaborations we found with MIT University now, with VIT since last 2 years in India, and more so in the US, both in New York and Connecticut. So I think that is my message. Pick a research area which is of current threat to our human health, and then find collaborations where you can do a comprehensive work which can be applied to mitigate the diseases. Thank you very much for listening to me.
Yes. Thanks a lot, Professor Parmar, for your illuminating talk on this detailed medicinal chemistry, and particularly the details of the viral and bacterial infections. You have given deep insight to us. Now, since we are running short of time, so I will allow only one question. Any one question, please.
Yeah, can you close the presentation so that we can?
Yeah, from the audience, young generation, please. This is the good opportunity for you people to interact with Professor Parmar.
I often come to Delhi, so I mean 3 or 4 times a year. I'm coming the whole January. I will be in Delhi in India, so we can interact if need be with any person who is of. I mean, you can write me an email, and we can fix a meeting.
Okay, since there are no questions raised, so we will again thank Professor Parmar for his detailed, illuminating talk.
Sir, I have one question.
Yeah.
Professor Verma, it was a highly enlightening talk. And the best part was the guidance which you have given to the youngsters about the ethics of collaboration. There was an emergence of black fungus during COVID, especially in India.
Oh, yes, yes, yes.
So did that also had to do something with antimicrobial resistance, or it was just a spread of an infection?
I think that also was in a way that the treatment was not given at a proper time, and the spread happened in addition to lungs, it happened in eyes, and many other areas traveled from there. So I don't think it is antimicrobial resistance there. It was more that the treatment was delayed. And then I mean, everywhere, I think the US, as we all understand and realize, is more developed. But number of deaths due to COVID in the US was 3 times than in India. So I think it has nothing to do with the AMR as far as COVID, the black fungus was concerned, or black. That thing, the nature of the COVID was concerned. It was more in my mind, I think more it was the delayed treatment.
Thank you, sir. Thanks a lot.
Okay. Thank you, sir.
Thank you very much. So I will leave the meeting now and wish all the people good health, and we will see you sometime in
One
future.
moment, sir. Excuse me. Sir, we from Deshbandhu College would thank you. First of all, thank you so much for your enlightening talk. And we would like to felicitate you with an e-memento. So kindly
I see that
accept
on
it.
the picture here. Yes.
Yeah. Thank you so
Thank
much,
you.
sir.
Thank you.
Thank you.
Maybe you can send it by email to me. I can keep a copy of this.
Thank you, sir.
Thank you so much. God bless all of you.
Thank you so much, sir. I was kind of listening you very, I would say patiently, and thank you so much. And uh, I think it's a dream of everybody to kind of work with you, collaborate with you. So, and you have, I think you have made my job very easy because I was able to correlate, when you're talking many things like, specializes and other AMR. So thank you so much. And I think I'm in waiting now. My next talk is there. So I definitely need your wishes, and you have taken this session to very, I would say, high place. So I don't know whether I can maintain that there or not, but I will try to do, sir. Thank you so much, sir. Thank you.
Thank you very much.
Thank you.
Okay, so now next is, okay. So now we move on to the next speaker. Dr. Ravindra.
Can I stay on maybe for a few minutes, or I should leave the meeting?
You can stay on, sir. No
Okay.
problem. Yeah. So our next talk is by Dr. Ravindra Vikramasinghe, and he will be talking mainly, his title will be Emerging Trends in the Development of Novel Therapeutic Modalities, Fostering the Future of Global Well-Being. So before he starts his lecture, I will just give a brief introduction. Dr. Ravindra received his MSc and PhD from Lucknow University in 1999, and then he joined Lupin Pharmaceutical as E3 grade scientist. In 2002, he joined GE India Technology Center in Bangalore. He joined Jubilant Life Sciences Noida in 2007 as a group leader. He joined Sigma Eldridge Chemicals Bangalore in 2010 as principal scientist, which was acquired by Merck in 2015, and presently is serving as its director and head of R&D Technology and Innovation Business in India. Dr. Ravindra is a passionate leader in life sciences and pharma sector. He has more than 27 years of experience in chemical industry. Dr. Ravindra promotes the new technologies and sustainability through automation and digital in R&D and manufacturing operations. Dr. Ravindra participated in many national and international conferences and symposiums as guest of honor, chaired scientific sessions, delivered plenary and keynote lectures. He has published several articles in peer-reviewed international journals and co-inventor in more than 15 patents. He is a recipient of several awards in his professional career, example, Industrial Scientist of the Year Award, Lifetime Achievement Award, Merck Science Award 2021, R&D Innovation of the Year, Impossible Possible Award, Innovation at Work, Management and Patent Awards. Dr. Ravindra is a member of Dream Team of chemistry experts at Merck, and he is Six Sigma Green Belt certified professional. Over to you, Professor Ravindra Vikramasinghe, please.
Thank you so much, sir, for a wonderful introduction. And I'm really very happy to be invited to this conference. And thank you, Dr. Pratibha. Thank you, ICHE, for inviting me for this wonderful session. And obviously, I was thrilled to hear that, Professor Parmar is going to be first speaker in this session. So, it's always a pleasure for me also to hear these stalwarts from the industry and, academia from India and obviously a globe. So thank you so much. I don't know how to share my screen. Can you make me presenter so that? Yeah, give me 1 minute. Yeah. Can you confirm that you are able to see my screen?
Yeah, it's very much visible.
Thank you so much. So as I told that, I was very excited when I got invited to talk in this conference, and particularly the topic of the conference, or the subject of the conference was very close to my heart, that human health and again the, let's say, environment. So both are the two areas where I think probably Merck and we all scientists, I think we are pushing forward. uh, and as I always feel proud to work with the Merck, which kind of, let's say, have a motto to kind of improve the human well-being all across the globe. So I'll not take much time, but I know we are already late here, and so
Yeah,
I'm
you
having
can
almost.
take 25 minutes. Yeah,
Oh,
no problem.
fantastic!
Yeah.
Excellent. That's great then.
Mm.
So what I did actually, I divided my uh, presentation in several kind of, let's say, parts. The first part will be about the Merck actually and what we are doing in human health, and the second, I think, I will connect to the topic what we have today. So the first topic about the Merck, who we are, and if you look at, just 1 minute. If you look at now, we are almost 21 billion euro company, which is a kind of largest in life science. Then we have a almost 60 plus thousand global employees, and the interesting part is that we are now 355-year-old company on Earth. We are the probably the most, I would say, historically established company which is still surviving. And I was very happy when Professor Parmar was talking that, making the collaboration is the first part, but the kind of sustaining the collaboration is the, very very important. So same here, that, running a company is important, but sustaining a company from 355 years is just something, remarkable. So I really feel very proud on that. Coming back to that, what are our main businesses? So I will not take much time on these slides, but if you look at, we are, I think, everywhere, where the chemistry is there and where is the human health. So it may, you can see that starting from the research, and then, in the lab, hospitals, and ultimately I think what we call is that, from discovery, from thinking process itself. Let's say we are thinking about bringing a new medicine in the market. So the genesis comes from the, let's say, researchers in the lab. So we support them, in their research in the beginning stage, even hypothesis stage itself, and then it goes to the commercial stage, and we bring the medicine to market. So I think that's a beauty. And we have three key businesses. One is the science and lab solutions. We have a process solution. We have life science services. So I'm going to talk much about a little bit more about life science services, but I'm also going to assure that I will not take much time on these slides, because these are the things which we talk, in many places, and some of the slides are also available, through our websites. So in the life science, services, what we do actually, we have interesting business called CTDMO services, and here C and T is a testing. Actually, if you look at CDMO, you will be hearing from many, suppliers or many companies, but when it's come to T is nothing but testing. So if you look at, the scientists when they make their, materials, so if you need a material in the large quantities, you can definitely approach the CDMOs partners, and they can manufacture those materials in the large quantities. But when it comes to testing, and today, if you look at today world, where the, the regulatory bodies, regulatory authorities are very very harsh on the, let's say, suppliers on and manufacturers on the testing and, regulatory documents. So here we comes in the testing. So we have a full flash service of CTDMO, and all across the globe. And this service, actually T part, we supply through the BioReliance contact testing service. If anybody having the more kind of interest about this, I can talk later as well, and they can reach out to me. I think my mail ID is already there, so, people can definitely reach out to me. And if you look at what the testing services involves, let's say starting from preclinical to clinical to commercial. So here the Merck, actually, we come all across here, and starting from your journey from discovery research and to the end. Interestingly, if you look at what are the novel modalities, when you're going to talk in the next slides, what I will be covering, actually not the monoclonal antibodies, but mostly I will be talking about antibody drug conjugate. HPA API is our another business, and the viral vector, and the mRNA and LNP. This definitely I will touch upon a little bit more, because you would have heard on Gandhi Jayanti 2nd October what happened. So I will not kind of disclose now, because you all are knowing that, but I think just kind of suspense you. So just wait for the next slide. So as I was telling, in our services, let's say preclinical to commercial. So we have, let's say, early phase development and process development services. We have clinical manufacturing, we have commercial manufacturing, and fill and finish. So what you're talking about, your clinical manufacturing, or anybody who want to do that, start from the, material in the large quantities, and then it goes to fill and finish. So like example, we are talking about the vaccines. So you will see the vial, you will get the vial. So that's what service we provide. And obviously testing, I already told that. And modalities as monoclonal antibodies, antibody drug conjugate, HPHP, high potent APIs, and vector vaccines, mRNA and lipid nanoparticles. That is also, I think we have full range of the novel therapies what is there now, and particularly in the currently in the market. Now coming back to why Merck, and why people have to reach out to us. I think I already told that we are servicing the industry, we are servicing the society from last 355 years, with a huge experience in the science and technology. And I think if you look at our mission statement, we call that we are collaborating with the global science and technology companies, and we are kind of solving their toughest problems. So if, let's say, a person or a scientist in the lab, he get stuck in the laboratory in the let's say research scale itself. So we are there, our scientists, our team of the people, they're helping them, designing their chemistry itself, we can be there. And then about then other phases of the, let's say, chemistry services, we are there. I think we always tell, from, let's say, concept to commercial, we are going always there, with our partners. This is very interesting thing, extensive regulatory, how to know how. So this is the one biggest challenge is coming to the market, that I already mentioned that regulatory part is a part which is, I think, going to be governed more, and is going to be more stringent now. So researchers in the lab, let's say professors, students, when they are talking about, I think I will definitely emphasize here that when you are planning your research, when you are kind of, let's say, phasing out your research, if you can include that element also, what can happen in the future. Is it going to be stuck because of some regulatory thing? Is it going to be stuck because of some, let's say, something which is something not planned, or which is something not know that going to be aligned to the regulatory thoughts? Yes, you may not be required to do in the, let's say, very early stage of your research, but yes, if keeping the mind, that is going to help the entire society and is entire the life cycle of the drug, if it is suppose something comes out very positive from your research. Next slide. As Ali told, we are kind of global technology leadership services we are having. We are kind of have very established supply chain management. We have a global capacity to reach. You heard that, you need a very big, I would say, lengthy chain, where, starting from x a point to z point. And proudly, I think we help our customers or anybody who want to collaborate with us in the entire life cycle of the value chain, I would say. Already I talked about, so I'll not take much time here. Now, I think this is very interesting slide, and this slide is talking all about what we are doing, in the, let's say, novel modalities area. Actually, monoclonal antibodies, that these are the fast growing, therapeutics, and probably I'm anticipating that the future is going to be lying here. And why, I will talk in the coming slide. Antibody drug conjugates, I think this is the very interesting, area, and which is kind of booming out now, I would say. And I think Professor Parmar has already touched upon that something, aspergillosis. What he had talked about aspergillosis, there's no cure. If there is a cure, there's only one medicine, a natural product like amphotericin B, or uh, um, the same other fungal diseases. So we have very few. But what is the problem? He mentioned already that they are toxic. They're highly toxic. What happened now when they go to body? They will not recognize other healthy cells, so they will kill everything. It's just like, you are having the missile, and you just switch on the button and it goes and kills everywhere, it goes and fall everywhere. So it's just like that. So I think this ADC is going to address the issue what Professor Parmar had just mentioned. I think I was very happily, listening him, and I mentioned that because I'm going to talk about that how this can be addressed. Now, high potent APIs, that today, you need APIs, you need drugs, medicines, but they are kind of, let's say, required in the larger quantities because bioavailability, their metabolism, there are many factors which come in the picture. But when it comes to HP API, they are very highly potent. So the quantities and their bioavailabilities and their metabolism is kind of very well established. So they have good efficacy and same time they have very good stability. Viral vector, you would have heard about viral vectors in the last couple of years when the vaccines came. So the viral vaccines, let's say COVID-19, I think that was one thing which has taught us a lot of new lessons. Actually, many people were not aware about what are the viral vectors and how the vaccine development happens. But now today in India, if you ask everybody in the villages, also people are talking about Covishield, Covaxin, Moderna, BioNTech, uh, and I don't know everything they talk about. I'm very, very happy about that. Coming back to mRNA and lipid formulation, I think this is I was telling, that this mRNA technology, this mRNA technique actually has just brought us a new area, which is going to be kind of the future. Trust me, this is going to be future of therapy. The reason, because you have, you know, that on 2nd October this month, two of the scientists, one is from the BioNTech and one from the University of Pennsylvania. They got a Nobel Prize for the discovery of mRNA vaccine. This, I think, Professor Karika. I have a slide here. So now this is about the sites what we have. I think we have the sites, manufacturing sites all across the globe. But this is very close to my heart because we have one in India, only one in India. That's in Bangalore, and we have our R&D center here in Bangalore, and along with the warehouse where I think whatever chemical any Indian, let's say, industry or any Indian academia are getting from Merck or Sigma Aldis, they're getting from Bangalore only. I already talked about ADC, and probably I'm going to spend more time on that. So this slide is just introductory slide, what services we have in the CTDMO. We just call it ADC. I'll not talk much here because this is going to be kind of a little bit more time taking. And the coming slide already, I'm going to talk more about that. So these are our licensed technologies in the antibody drug conjugation area. ADC Express, we have Cheeto sensor technology, we have Safe Bridge certification technology. So all these technologies are available for the global researchers, whether in industry or academia. So people who are kind of working in ADC or people who are planning to work in ADCs, they will have access to these technologies. And trust me, these technologies are going to be kind of driving the future of the ADCs. mRNA and lipid nanoformulation, as I told you that the world is changing, and mRNA is something has given a new access to the, I would say, the globe, and new access to the people who are working in the research and development and manufacturing are kind of, let's say, human health, a new tool, I would say, which is something, uh, it just, I would say, remarkable, and it's historical, I would say, if I have to summarize that. The reason being because it is not that mRNA is new, it is known from the decades. But what happened now during this COVID time, this discovery and the particularly the push on the mRNA vaccine and as well as the drugs, it has just changed the entire world. So if I have to talk today, the Merck itself, I think we have entirely different let's say business which is kind of addressing mRNA and LNP. And I'm proudly telling you that my Bangalore R&D team is working on these areas, mRNA and LNP. We are making a lot of novel lipids. We are already supporting, let's say, our RNA drug development or vaccine development programs, which is running globally. Now I will come to this slide, and earlier what you heard, it is just about the Merck, what we offer. Now I'm connecting this to today discussion and what, let's say, theme of the conference about the future global well-being. Uh, so if you look at novel chemical modalities, there are many, there are several, but I think one already I talked about, mRNA, and then we have a PROTAC, protein degrader building blocks. So this is another very interesting technology which kind of, say, uh, is in the market, but the problem is that we don't have any drug. But very soon you will have one drug, or maybe you're going to have many drugs in this technology. I will talk a little bit about this protein degradation technology, and then I think I plan to talk about antibody drug conjugate, which is, I think, is called as a magic bullet, and is this going to take over probably all the kind of existing, I would say, therapies. May not be in the very soon, maybe not in 5 to 10 years, but definitely in the going future, you will see that mRNA and ADCs are going to take over the entire healthcare system, and they are going to be the leading. You may be hearing that APIs, pharma, but now after a few decades you will hear only about ADC pharma, or let's say mRNA pharma, mRNA, medicines only, mRNA drugs only. So that's the future, and you have to be aware with me. mRNA, I already talked about that. And here the slide, which I think probably I have to really, we all have to congratulate, Professor Kariko and Professor Wisman, both these guys, Professor Drew, actually he's a professor in the University of Pennsylvania, and Professor Kariko, uh, she is a professor, and president of the BioNTech. So these two guys, I think they have worked on extensively on the mRNA vaccine for the COVID-19, and this has resulted in the Nobel Prize for this year. And this is very well deserved. Now, I think I don't want to spend much time on that, this area, because this is available, in the Nobel Prize Committee website. You can see this picture. This is very simple. If you look at unmodified mRNA, and then they modify the base, modified RNA, and then it goes and kill your pathogen. You can see here in inflammatory response. How they do that is very simple. This mRNA, this goes in body, it doesn't enter in the cell, let's say pathogen cell. What they do actually, they just replicate the let's say pathogen protein. So let's say in the our body, we have, let's say, immune response system, and we have cells which are responsible for fighting the diseases. What happened now when any foreign pathogen, or let's say virus, comes inside the body, the self, the response or immune response get activated. What happened now, it takes time. So in this mRNA case, what happened now, this mRNA goes inside and it's kind of, we call it the blueprint. It creates the blueprint of the pathogen, the protein, and then the immune response in the human body, it gets activated. So that's how this works. Good news is that safety and efficacy data of mRNA based therapies in human through COVID-19 vaccine has opened up a new door of the development in this area. And particularly, this is a my line actually, which I already posted in the LinkedIn, that this is going to lead, and this is going to open up new opportunity for mRNA based drugs. It is not only the vaccine, you will see the drugs which are going to be based on the mRNA. Happily, I'm informing all of you that Merck, we have just, even before knowing this Nobel Prize, before October, we have just opened two new R&D manufacturing units in the Germany and US, and we spend almost 1 billion euro. We have just spent 1 billion euro this year on this mRNA manufacturing technology. I think this is definitely a proud moment for everyone, and the reason, because this is going to be helping the entire healthcare system of globe. It is not about India, is not about the Germany. So whatever outcome is coming from these facility which Merck has spent, it's going to be serving entire globe. So this is, I think this is very important, very close slide, and picture, which is kind of, I always feel happy to talk about that. What we are doing actually, if you look at, this is our recent patent, what you have seen, Protax. So I think this is, you can see that this came in the June. I got this in the month of August only, and this is the just latest, very latest, last week we got that, and this is on the antibody drug conjugate. So these are the two recent patents, and as as Pramar sir has talked about that collaboration. So you can see the collaboration. This is a global collaboration from India. I think I'm obviously the inventor, and here in the this is a global here also. I think you can see me here. So not only me, my team also there. So this is a global patent which has just published, and interestingly, why this is very important, because these two are the close to the the what is going on the new modalities. So this one, the left right hand side, is about the antibody drug conjugate, and the first one what you're seeing this left hand side, it is about protect technology. So we are just making a lot of linkers which are going for the protect use. So I will just touch what is a protect. It is nothing but proteolysis targeting chimeras. uh, and if you look at the just you type in the Google browser, and you can see that a lot of literature will come in front of you, and this is one which is very close to me, and I just taken the picture. Protein solving drugs could be a next blockbuster therapies. So this is just literature popularity. So I just captured here so that you will be aware that what is going on. As I can see here, this 2001 and 2, there were only few, maybe one or two literature references were there on Protax technology, but going forward in 2018 it became 968, and in 2023 if you look at almost 1,000, more than 1,000 publications are there. So this area has gained a momentum, and a lot of research groups across the globe, they're working in this Protac technology. So,
Excuse me, Dr. Ravindra, just couple of minutes left.
okay, sure. Thank you. Thank you so much. I will just rush now. So these are the two molecules which are in clinical trial. One is called ARB-110, and the other one is ARB-471. ARB-110 is androgen receptor. This is for the, let's say, prostate cancer, and the ARB-471 is for breast cancer, estrogen receptor. And this is just, you can see that which area and how many targets are there. So there are total, 54 drugs which are in the trial on the Protax. I'll not take much time here. This is a Protect journey started in 2001. In 2022, two drugs, ARV-110. This company called Arvinas, they went in the clinical trial, and still I think they are under clinical trial. So I think we don't have, I'm sorry, we don't have any medicine in the market on the Protect. And however, 71 Protects are in the different phase of clinical developments. I'll not spend much time here now. What we do in Bangalore, so you can see this is the kind of, let's say, protein of interest ligand, and this is a half protect. This is a half protect, and when you connect this protein of interest ligand with the half protect, we call it the partial protect. With the linker and with the ligand, you get the full protect. This is a drug actually, full protect. But however, this is the active part which kind of goes and interact with the cell. So in Bangalore, what we have done actually, we have supplied almost entire globe with almost 500, 400 to 500, this kind of molecules. And the patent what I have just disclosed here, it is about making the novel linkers uh, for the product. Now I'm coming back to antibody drug conjugate. And what are the antibody drug conjugate? Obviously, this picture is taken from the our website. So here, I think there's only one difference. When I think Parmar sir was talking about aspergillosis, I was listening carefully. Is that when you take this medicine, what happened now? It kills all the cells, healthy cells and the disease cells. So there's no kind of recognition which is bad, which is good. Whereas the ADC, antibody drug conjugate, is just, I would say, guided missile. It is a guided missile. It will kill only the disease cell. It will not kill the healthy cells. This is the whole concept of the ADC. And I'm sorry, I'm rushing now because time is less, but you can leave that. You can definitely read out these things from the our website, Merck website, or even from the open literature. So it is not that ADCs are very, very new terminologies. It was used by Professor Paul in the 1913, and he has first coined the word magic bullet, which is kind of, let's say, it is going and killing the only the disease cell, not the any other thing. So you can see here 2013, the journey continued, but the most of the research happened after 2011, I would say, after 11, and is happening now. So total now we have almost 13 ADCs, antibody conjugates, approved by FDA. Whereas in the Protec, I think I told we don't have anything. 71 are in the clinical trial, but in the ADCs we have now 13 drugs already approved by FDA, and we have almost 111 actually this whole number, this is on a website. So we have 111 actually, which are in clinical trials. I'm rushing. How this is the kind of how the ADC look like. If you look at this is antibody, monoclonal antibody from the disease cell, the body, and if you look at the linker, this is the drug actually. This is here payload. We call it the drug as a payload, and you link this payload with the linker, and this linker will have a site here which will attach to the monoclonal antibody. Again here, popular literature popularity, if you look at every day, thousands of researchers coming in this area, and these are the couple of few selected literature references which I thought to help to put here, so that you all can have uh, uh, excitement about reading this. And particularly when I talk to any conference where I see a lot of researchers and students and the professors, I definitely want, and I wish they should focus, shift their focus to this area, because always what happen, we are too late. When everybody going to stop, then we start. Whereas in this area, these are the two area which kind of have just gained the momentum, and we have to jump in this area. So this is just example. And again, you can see the patent, and this is the patent landscape. This is very important slide, very, very important slide. If you look at here, here's right hand side data. If you just look at a SciFinder search, antibody drug conjugate you type, you'll get almost 20,000 plus references. And out of these 25,000 20,000 references, you will have around 15,000 journals references. You have almost 5,000 patents and 3,500 reviews. These patents, what I'm talking about, 5,000 patents until now, okay, and 2023, if you look at here. Whereas what is the projection actually? Market, this is nothing but a market projection. It is just projecting by 2032, we'll have almost, let's say, 15,000 patents only, or more than that patents on the antibody drug conjugate. So it means, you are getting a visibility here, how this market is going to be turning up, how this area is going to turning up in the future. That's the reason I was telling this is going to lead the future of human health. Interestingly, I noted another point. If you just put in a Google, you just put antibody drug conjugate, within 30.38 seconds, I got 31 million hits actually. So this was very exciting to me. This is very, very exciting to me. Again, same thing. This is if I don't correlate the market, how the market is going to respond. If you look at here, today 2023, the ADC market, antibody drug conjugate market, is around $8.5 billion. $8.5 billion market. But the projection is that by 2032, this market is going to be $35 billion. $35 billion, almost 5 times or 6 times more. And this is the CAGR, compounded annual growth rate, 17.5%. Trust me, we always tell, if you are having the low double digit, 10%, 11%, that is fine. But this market is going to grow in 17.5, percent CAGR. That is the remarkable. What are the area you look at here? The major research is happening: breast cancer, blood cancer, urethral cancer, and other cancers. And these are the the revenue generated by the most of the medicines. And this is Kadcyla, it's one of the more medicine which is generating a lot of revenue. And these are the medicines which are already, I think you heard, the 13 approved out of 13. I think this is the one blockbuster which is generating more revenue. This is
Excuse me, Dr.
structure.
Ravindra,
I'm
time is
done.
over.
I'm done.
Okay.
I just came to last.
Okay. Thanks. Yeah.
So yeah, these are some linker payloads. So I think I'm not going to take you much about the chemistry. So I will just skip how they are getting made. So this is nothing but you have antibody conjugate. You can see here, and this is your linker, and this this payload. So they get linked with melamide actually. If you look at the bond, so this is your complete ADC. How this makes, I think it is already available, but you can review these slides, and I'm available. Anybody want to know more about that? Because in Bangalore, we are in India, we can help and support if anybody want to plan any work on that. This slide, definitely I need maybe a 30-second to explain you what we did actually in the Merck Bangalore. So the patent what I have just shown you, antibody drug conjugate, is about the PBD core actually. This is the PBD, dimer is the toxin actually. And what we are this is our technology, the licensed technology. We offer these materials through our websites. People can go and buy, and they can attach this to toxins, like, you heard the amphotericin B. If you attach this with amphotericin B, and you can deliver to the disease cell. This is very interesting. And if you anybody, let's say, making the medicine on drug, and particularly antibody drug conjugate, is it traditionally you have to, let's say, go back with the 10 to 15 step of the synthesis. Whereas if you take these material which are already available with the Merck, you have to just maybe do 1 or 2 step, 2 or 3 step maximum, and you get your the active drug. With this, I came to the end, and thank you so much. This is a beautiful site of the Merck in India. This is our corporate block, and this is our laboratory area. This is our warehouse. This is a green site. We have almost 40% energy we are generating from the solar cells. Uh, so thank you so much, everybody. And definitely, I would like to thank my leadership team and my colleagues in the Merck India. Obviously, the entire organizing committee of ICHE, Dr. Pratibha, head of department, and one of my senior, my colleague, my friend, Professor Divan Singh Rawat, who was in Delhi University. I have a lot of friends still in Delhi University, so I have to acknowledge them also, because we are connected. And whenever they see any opportunity, they always interact with me. They always inform me, I think, and then I get invitation, and I feel happy, because whenever opportunity comes to interact with the academia, I think I really feel very excited. So thank you all, thank you so very much, and for listening me. uh, Thank you so much.
And yeah, thanks a lot, Dr. Ravindra, for giving your, insights on the development of novel therapeutic modalities. I'm sure there would be a lot of questions, but since we are running short of time, so I will allow only one question. So the
Please.
session is open for question. Any one question?
Dr. Ravindra, it was a very nice talk. Especially, it gives an overview of what industry is doing to the youngsters. And you also made us aware about the latest drug developments related to PROTEC, antibody drug conjugates, and mRNA. A very quick question. So during COVID, we have a different type of vaccines, including the mRNA, RNA, as well as the conventional heat inactivated viruses.
Right.
Is there any study which shows which one was more efficacious, even though mRNA discovery is quite a good development, but is there any study?
Yes, there are a lot of study conducted, and that's the inactivated virus. This is a tested, proven, and the historically established concept. So I think that is already, I think there's no debate on that, because that's easy. What happened now? I will tell about this mRNA. That BioNTech and the Pfizer, Moderna vaccines, they are mostly mRNA vaccines. The advantage of these two vaccines are that uh, they are working with a targeted delivery module, actually, mode, sorry, mode. So they have a lot of advantages. First thing. The second thing, the cost also is coming, here cost advantage is coming huge, and efficacy is high. So to be honest to you, this conventional historical vaccine, they are not going to out of the market, but they are going to stay, and they may be more in the in the market. But whereas this mRNA and these vaccines are coming, or therapy, they are definitely going to take the space which is already there in the, let's say, traditional therapies. Okay, very interesting. It is not about the only vaccine. Sorry if I have just only made this statement about vaccine. What happened now? mRNA therapies are going to be therapy, and they are going to be leading in the pharmaceutical also. You will have the mRNA drugs also, the, let's say, pills, what you're going to take that. So the one thing. Second thing, it is not limited to the virus only. Interestingly, it is not going to limited to virus only. It will be, and it is not going to limited to the cancer. You have seen that I was talking about mostly oncology targets, whereas this mRNA is going to be expanding to the, let's say, malaria. I don't know whether you heard or not. The malaria vaccine also coming with mRNA. I think you heard with Professor Parmar, AMR, right? Antibody resistance. What happened now when you have mRNA vaccine for malaria, when you have mRNA vaccine for tuberculosis, which is kind of very very high now, it's coming in the market. So mRNA has opened up a new space where I think probably mRNA, you have, they say, antibody resistant. So this is going to kind of changing the world. So this huge work is going to be coming in the future, and again, sorry, it's not only oncology, it is going to be every area. I talked about aspergillosis. You have seen that. I noted that. I think maybe the fungal resistance, there also mRNA is going to work. So
Thank you.
to be precise to your question, study done already, and both are going to stay in the market, whereas mRNA will find more space. And it's not only in the vaccine, but in the other traditional drug therapies.
Thanks a lot. Thank you, Dr.
Thank
Ravind.
you. Thank you.
Dr.
Yeah.
Ravind, just a moment.
Thank you so much, Dr. Singh, for such an interesting talk. And as a token of respect, kindly accept the e-memento from Deshbandhu College.
Thank you. Thank you so
Displayed
much.
on the screen.
I'm
Thank
happy
you.
about that. Sure. Thank you so much.
Thank you
And
so much.
now
Thank you.
we move on to the third talk, that is by Dr. Anthony Atlagata, and he's working in ICT Hyderabad. Before he starts his lecture, I would like to give his brief introduction. Dr. Anthony Atlagata has obtained his bachelor's degree with specialization in industrial chemistry from Osmania University, master's and the PhD degrees in chemistry from the School of Chemistry, University of Hyderabad. He has worked as a senior scientist at the Department of Energy, Pacific Northwestern National Laboratory, USA, before joining Indian Institute of Chemical Technology, Hyderabad. He was awarded with Bionsky International Fellowship from the Polish Academy of Sciences, Poland, and Ramanujan Fellowship by the Department of Science and Technology, New Delhi. He has published over 55 research articles and a book chapter. His research interest is to understand the chemical principles and biological functions and apply it to human health and biotechnology. His technical expertise includes chemistry, biochemistry, protein engineering, molecular structure, cellular and computational biology, bioinformatics, structure-based drug discovery, and biotechnology. He also serves as an inspector on behalf of the Government of India for companies that adopt good laboratory practice. So over to Dr. Anthony.
So, do my duties for thanking the organizers and very specifically Dr. Pratibha and Dr. Divan Rao, who is my friend also in the Delhi University, for recommending me and tuning me here. I'm very happy to be here to discuss about what we have recently done.
Yeah.
सर कैसे लगाते हैं इसको?
Just
वो तो है मगर
Sit and
No, I think it's okay.
because from the camera it is
Oh, is
from
it?
the back side.
Ah, okay, then I think I'll stand here and then give. Okay, then I have to stand here. Yep. Thank you again. So we have recently published this paper in a journal with the impact factor 8. So here the question here is, we have heard about the drug resistance, right? It's a drug resistance. And how many of you remember COVID time? Sara Lecturu, you still remember, right? And a lot of news everywhere and no classes for TV. Huh? So some good, some bad, and for some experience good things, some experience very bad things, unfortunately. And during that time, we have seen different versions of COVID, right? पहले तो वाइल्ड टाइप बोला, बाद में alpha बोला, बीटा बोला, gamma बोला, delta बोला, पता नहीं कितना. And there are hundreds and thousands of versions of it. What do they mean? What these versions means? क्या होता है उसमें? Why? Huh? Mutations होता है. Yeah. So this mRNA is not. This is mRNA virus. Okay. Again, I don't want to repeat what Dr. said, but yeah, it's mRNA virus, and then there is a enzyme called RNA-dependent RNA polymerase, which is, does not have a real proofreading, and it is nature made it that way, so that it has got freedom of letting some mutations to happen. And most of the time, 99% of mutations will not survive. Though they will survive basically. And that's the reason why we cannot easily make drugs. And even if we make a drug, it's going to be in trouble. That's the reason why, HIV, you know that word, right? HIV, it's been 50-year-old disease, and still we don't have a proper drug. Of course, I want to use a word called combination of drugs. So they give several drugs. The patient suffering from the HIV will have to take combination of drugs, not only to cure the HIV, but also with the associated disease like tuberculosis. So today I will tell you a little bit. My chemistry is very simple. and biology also very simple. I have only 10 slides. So if you focus for next 10 minutes or 15 minutes, I think it will be over. Okay. So so it's a what is the drug resistance there? So basically, drug resistance is, suppose you look at sometimes when you give a talk, something हाथ में कुछ रहना चाहिए, तो problem हो जाता है, नहीं तो you feel you are helpless. Thank you. So basically, if you look at this, so this is a general pool of virus, basically mostly of the same one type that we call wild type, and then there are some random mutations that are there. Then at this time, if you are giving a drug, then what happens? The drug actually will target all of them, but suddenly one or two fellows will survive because this drug could actually find a resistance. That means that that viral actually have some mutation where your drug is targeting. So generally, what does drugs do? They go bind to a specific protein. For example, say protease or an RNA-dependent RNA polymerase. So they'll go and bind. But suddenly, if the mutation actually is there, then this drug may not be effective in binding to that mutant form of the virus. Then that survives. And then once it survives, then it starts. So first you see that infection really goes down. We feel very happy and we are happy. And suddenly, after 2 days, you see again, you get fever and then we are seeing this problem. So then you slowly see this increasing. And then now again, new mutations are also coming up, which are also drug resistance, basically. So that's the problem where we are facing with the viruses, basically. So if you look at this, drug resistance occurs faster than the drug development. It is true. So 1944, the first drug antibiotic was invented. 45, we have drug resistance. For just 1 year, we have drug resistance. And we didn't have antibiotics until the time. Just the whole world was waiting. The world was fighting and the wounds and disease, and we didn't have antibiotics. 44, a miracle drug has come, and suddenly 45, we have a drug resistance. It's bound to happen, whether you systematically use your drugs or doctors control availability of drugs, you still have. This is the nature of the disease, nature of the bugs, what that is there. So how to overcome the drug resistance? As I told you again, it's not easy, but again, now being scientists, we have to have all jobs. All of you also are looking for jobs. आपका BSc होगा, MSc होगा, तो अभी जो, so what are you? So we have to propose some of these things. हम कैसा? How we stop the disease? So you propose something, get some funding, get some job, and start doing our science. So here, how to overcome the drug resistance, and then multiple drugs. I told you people give multiple drugs. So multiple drugs are targeting not one protein but two proteins. Say we have a drug for one protein, say protease, and another drug for say another RNA-dependent RNA polymerase. I'm talking specifically about the virus, basically. So, or we can also have dual inhibitors. What is the meaning of dual inhibitor? Same drug, only one molecule inhibiting two enzymes. Suppose in this first case, so we have a molecule targeting two proteins, and then when we find one protein finding mutations to the drug, then the wild type is killed. But if it is a dual drug, it can also go and kill this also, basically. So when the say probability of mutations is there, then when you have a molecule inhibiting all possible enzymes, then the survival of the virus is less actually. That way we can overcome the drug resistant microbes, or it could be bacteria, it could be virus, basically. So this is what we have done, and very simple chemistry. So again, this is just my lab, what we do. And if you have heard, I know it's the third day, Sunday morning, focus karna thoda mushkil padta hai. So I understand that. So I'm a chemist. I studied like you. I did my industrial chemistry, BSc, mathematics, chemistry, industrial chemistry. Yeah. And then I did my PhD, MSc and PhD chemistry. I never had biology until my PhD. Then I decided that I have to apply whatever chemistry I've learned into biology. Chemist being is very advantageous. You can think a lot of beautiful things in biology, where unfortunately biologists have little difficulty in understanding the molecular structures, basically, in terms of atoms. So we use, being a chemist, I became a protein chemist and protein structural biologist. We solve a lot of protein structures using X-ray crystallography. And then we use proteins for various applications, industrial applications. You can use diagnosis and pharmacology and infectious disease. So if you just go to the SARS-CoV-2 virus, so there are 3 drug targets. One is RNA-dependent RNA polymerase, where there are some drugs that are already available here. So this, what it makes is, it makes takes the RNA copy that comes into the cell, And then this protein actually keeps on making new photocopies, making new copies of the RNA basically. And then it repacks and sends it back. Then these two proteases are there. So these two proteases is called 3CL-Pro or main protease we call, and there is a PL, papain-like protease. And these proteases are required because when the virus RNA gets inside the cell, then a protein is made. And the protein actually is, because this has got about 18 proteins are required for the function of this virus. Then, but it's all made in one single polypeptide. That is not functional and that is not stable as well. So basically, please. So basically, then you have your proteases. The protein actually coded within the sequence. It actually, suppose this is a long ribbon of polypeptide. This protein folds itself and then comes back and cuts itself basically, and then releases a protein to be functional. So that's what these two proteins does. So then we want to target one molecule targeting these two proteins because they have similar chemistry but not same structures in the active site. The chemistry, they have cysteine proteases. Cysteine is having sulfur. Sulfur can become a nucleophile under appropriate conditions. Typical cysteine pH is around 7.3 or 8. But when cysteine protease, the pK actually reduced to 2.3 or 2.5. So it's very nucleophilic. So that means that S is always minus. That means it can actually attack the electrophiles. So that is the principle what we have taken. That's the only principle what we have taken. And then we designed our molecules. So this is, suppose it is M-Pro or 3CL-Pro. You can see the active site is little deeper and bigger. And this active site is very, very shallow. There is little scope for inhibitors to go and bind, but this possibility is there. But if you look at that, both of them actually having the cysteine sitting in the middle. That's what they do the chemistry on the peptide hydrolysis. So this is little more into deep. So as I told you, mutations could happen anywhere here. So your drug cannot bind. Suppose if my molecule targets both the proteins, one of them, even if undergoes a mutation, then this can be inhibited, so virus can die. So this is the concept. And so this, again, very simple molecule. This is a chloroquine. So two chloroquine. So it's a not chloroquine, but a quinine moiety here. So, and then what we have done is, because this is a nucleophile, we want to create electrophiles in the molecule. And electrophiles, because they have two different active sites, we don't want to keep one electrophile, we want to keep two electrophiles. So that no molecule can slide depending upon the active site shape, can slide. So we have one electrophile here because it's a 2-chloroquinolone. So actually a nucleophile can come attack on this one and the chlorine can leave. So can have a covalent bond. And we also can have a imine. And we designed this based also on the simple synthesis required. Suppose we take this molecule in the form of an aldehyde, and then we take this as an amine. So just condense them, simple chemistry. We condense them, make an imine. Very simple. You can make as many number of molecules basically. So we have taken this. And all this we have done during COVID time. While the COVID is still the Delta or Alpha, when they were really furious, they were working in the lab. So this is a chemistry. I told you very simple chemistry. And then we also require proteins. So we have cloned both the proteins in the lab. We expressed them and purified them. And then we got the peptide-based substrates. And then we established the kinetics. So you can see the both PL-Pro and M-Pro with the kind of KM, KCAT, because these are important to establish any enzyme inhibition. Once we have this shows that enzyme is active, and we use these parameters also for the inhibition studies, basically. So this is my collaborator who made chemistry. We designed the molecules, made chemistry, and we made this 10 molecules. So look at all of them have this chloroquine, just this small substitution on the left or right. And then we have on the right, we have this imine group here. So the slight variations. So you can see the slight variations are made on the right side, on the top here. So we tested for activity, expecting that all these molecules should inhibit both the enzymes. And then what we see is that the molecules C3, C4, C5 inhibit 100% at 100 micromolar. It's a little high concentration, but only one enzyme they're inhibiting. And can you minimize this fellow? Because I want to show the structure. Anyway, so these inhibit only the M-Pro, only one enzyme. Yeah. So these inhibit only one enzyme, and then, but that is not what you expected. We want them to inhibit both the enzymes, but only one is doing. We took the Then we have compound number 10. The same thing, ज़्यादा कुछ फर्क नहीं है. You have the same amine and then slight longer modification. But now we see this molecule inhibiting both enzymes, 100%. And just to tell you, there is in the literature so far, the design of dual inhibitors is not there. Serendipitously, people identify a molecule inhibiting both enzymes, but medicinal chemist, first people to report that designing from beginning, we design molecules to inhibit both the enzymes. Again, so we just took a 100% inhibition of both the enzymes. Then we have done analysis how these molecules are inhibiting. So we made 2 electrophiles, right? We should know what is actually the reason why they are inhibited. So we took all these 4 molecules, C3 to C5 and then C10, and then we did the enzyme kinetics. You can see, if you can understand enzyme kinetics in terms of numbers, you can see 1 micromole or 2 micromoles, 2 micromoles, 2 micromoles, and about 1 micromole. So that means the Ki is very good. In fact, to start with simple chemistry, we have achieved a very good inhibition. And particularly look at this molecule C10 inhibiting both the enzymes at 1 micromole. So then we want to understand what is the mode of inhibition, the mechanism. So we have incubated these molecules with the enzyme and did the mass spec. So this is the molecular weight of the enzyme, and then we can see increase in the protein size with the molecule corresponding to the one of the molecules basically. When these three C3 to C5, we see this covalent modification. That's what we designed, right? And then that's what they're also doing here. So, and then we look at the molecular weight comparison, we can see that this chlorine actually is lost. So this is where we see a covalent bond formation here. So these 3 molecules are inhibiting this only M-pro, you can see only M-pro covalently. So then I told you that the C3 inhibiting covalently, and then you can see the molecular weight, and you can see where the activity is done. Then we went to this molecule C10, which is inhibiting both the enzymes, and very surprising to us, we did not see any covalent bond formation basically, either both the enzymes. So that means this molecule actually binding non-covalently to both the enzymes. And so we see only the parent protein molecular weight. So we don't see any increase in the molecular weight. That means that we have a dual inhibitor that inhibits non-covalently. So what is the advantage of being covalent, non-covalent? I'll show you the data, what we have. So, but before that, what we have done is, so we said, since anyway this chlorine actually is critical for binding, then we said, okay, let us actually modify this imine, because imine is easy to modify. So we said that let us do very simple chemistry again. So we have taken this molecule number 10, and then we converted it into a 4-member ring and a 5-member ring, single step, very single step. Again, we did the activities. Now you see the beauty of it here. So this is the original molecule C10 inhibiting both the enzymes at 1.3, 1.6, and 1.2. Now you see in the nanomolar range, suddenly the simple modification we could actually improve by 10-fold, and getting into nanomolar is very, very important actually. So we got this C11 inhibiting at nanomolar range, and then C12 again. Now this is going up. That means that this is where is the optimized structure for binding to both the enzymes. Then now we will take this molecule and then do the modifications here and modifications here. So if we can actually further improve the efficacy. So all molecules bind non-covalently, that means no increase in the molecular weight. So we have to see then toxicity, right? At the end of the day, molecule will have to test for toxicity. So we have taken this all molecules. So you can see. So there are 6 slides, but 6 in particular. You can see vero cells. This is the epithelial cells in the lungs where we see the infection happening by the virus. So we have to check these molecules are not toxic to those particular cells. If they kill the cells, then there's no way that we can use them. But if you look at this molecule number 3, 4, 5, very toxic. They're covalent, right? They have a good electrophile. In the cell, there could be a lot of nucleophiles are there. They can go and bind to any protein without any selectivity, and they can kill the cells. That's what they are doing here. But when you come to this compound number 10, 11, and 12, even up to 100 micromoles, we don't see any toxicity. This is what is required. So that's why people don't like having covalent inhibitors, because they can be very random, and then they can go and kill all the other proteins and kill the cells as well. So now we got, again, I will not go through all these things. These are the numbers, but you can see all these 3 molecules that we made are non-covalent and non-toxic. So now we made it. So we have to also look at how the mode of binding is. So simply we did little bit of, again, we did a lot of mutations and then structures basically. I don't want to discuss now. So basically what we see is that these molecules, those compounds 3, 4, and 5 are binding covalently to the active site cysteine here, whereby throwing away the chlorine, and then they're binding covalently here. And then the non-covalent molecules actually are binding close to the cysteine, but they actually are not inhibiting the binding to the covalent. They're close, but not really the electrophile actually is not close to that one. So now we even removed the chlorine also. We got some better data now. I am not discussing here. So finally, the conclusions is initiated with covalent inhibition design, then developed non-covalent, non-cytotoxic dual inhibitors against Tempro and PL-Pro. So this is a group where I could be seeing some of you sitting here. Yeah, you can come. Okay. So these are the 3, all girls have done this job. Okay. Okay. So open for questions. Thank you. Section is open for questions. Which we are modified from imine, right? Fusion of other yeah, other ring, right? Chlorine and right. So, efficiency is probably fixing the conformation of the molecule. See, basically, if you look at this and this, they are almost isostructural. There could be little bulkiness is there, but otherwise, see, here in this case, we may actually have both cis and trans, but here we are picking up only one. This chemistry can work only with the cis conformation, not with the trans. So probably we have only cis conformer actually is active, not the trans conformer. So that means that we actually are not only removing the trans conformer, which is not required by doing this chemistry, and we also are able to achieve the fixed conformation, because the larger number of torsion angles, then larger number of orientations what a molecule can fit, and then it requires more energy to minimize the active site. So we have minimized that. If you look at your the entropy part, basically. So the chlorine and the two groups would be rolling in attaching themselves to some residues in the active site. See, basically, this is not really playing great role here, but this actually is fixing the positions of these fellows to fit into the pockets. So this is not really playing great role. This is only fixing it. That means the orientation is now locked now. Yeah.
Sir, I have one question. Like I think in the next slide where you are showing the toxicity graph. So what is that lead compounds? Toxicity data of lead compounds. So what is
lead compounds?
Oh, so it's lead.
Lead
okay.
compounds. Yeah. Yeah.
Okay. Thank you.
Haan ji. Yeah. What cocktail now? No. Only one molecule. See, this molecule is doing the dual inhibition. One molecule inhibiting both the enzymes at a given time. It's a dual inhibition. Yes. Yeah. So I thank Dr. Anthony for his
Thank you so much, sir, for such an interesting and a crisp talk. I would now request Dr. Sharma to felicitate Dr. Anthony with a memento as a token of respect and appreciation from our side.
Thank you.
Ma'am, please.
You both are in CSR.
Very nice.
Oh, very good. You do activities?
Yes.
And then doing the
Yes.
Yeah. Thank you.
Very nice.
Thank you.
Ma'am, please.
So we now go to the last speaker. It's actually a video presentation from Professor Maria L. Pereira. She is the professor in the Department of Medical Sciences at University of Aveiro, Portugal. And her title of presentation is Nanoplastics and Global Health Concerns and Future Goals. Before we start with her presentation, I would like to give a brief introduction about her. Professor Maria, actually, she is a PhD in biology by the University of Aveiro and is working as an associate professor with Habilitation Center at Department of Medical Sciences of UA. She has 40 years of teaching experience and research in histology and cell biology. Her research interests are adverse effects of pesticides, nanomaterials, and drugs, which has been conducted as a member of the Associate Laboratory CICE-ECO, Aveiro Institute of Materials. As a member of the World Union for Herbal Drug Discovery, she studied on natural products and with biomedical applications. She was the president of the Portuguese Society of Electron Microscopy and Cell Biology in 2004, director of the Master in Molecular and Cellular Biology at UA in 2008 to 2018, and fellow of the Royal Society of Medicine London in 2021, being a member of the Pathology Council as well. She published several papers in international journals, collaborates as editor, and has recognized several international conferences. Recently, she was distinguished with 2 Pedagogical Innovation Awards in co-authorship and received the Dr. Matthew Elcher Memorial Award Research Fellow by the Asia Pacific Consortium of Researchers and Educators in 2022. So now we start with her lecture of Professor Maria.
To all of you. And first of all, I would like to thank Professor Pratibha Kumari for the invitation for this interesting conference. I'd like to wish you all a very pleasant talk, and it is an honor to be present to share some of my results with you. In this talk, I will speak about nanoplastics and global health, and I will focus on some concerns and future goals. I'm from University of Aveiro, Portugal, and I'm based at the Department of Medical Science. During these last years, I have been still studying the role of heavy metal, some pharmaceutical, pesticide, and microplastics as emerging contaminants on the public health. And some studies and some research has been carried out using several models, in vivo models, live cells, and the in vitro models and in vivo models by rats or mice. So in this talk, I will focus on some emerging contaminants, namely some nanoplastics. I will focus on the adverse effects on health, and I will show you some results from research and also future goals. As we can see, emerging contaminants are organic, pseudo-persistent, and unregulated new contaminants in environment, both water, wastewater, in trace concentration. And in this list, we can include some antibiotics, some pharmaceuticals, such as antidepressants, beta blockers, for example, some hormone, endocrine disruptors, industrial additives, and microplastic, and also nanomaterial. They are released to the municipal, industrial, and domestic wastewater, and is the vehicle for the aquatic environment. You can see several books pointing how potential and other of these chemicals. Then we can see the origin of these chemicals. They are from industry, they can come from hospital, through medicines, then houses, and these chemicals can go through effluents from industries, livestock treatment, human waste, and personal care products. You can see a big threat on different ways how to be contaminated. And personal hygiene products like sunscreen, cosmetics, and other products like pesticides, hormones, as I have mentioned before, solvents, and packaged foods may be origin. Human exposure to plastics maybe from bottled water, for example, we are using, but it can come from air pollution and also to the diet food, some dust and tea from tea bags. So we can say that there is different ways of contamination. But the contamination of nanoplastics and microplastics, it depends also on the size, and it also depends on the shape and structure, the color, and it can be of different origin. For example, there are spheres, irregular particles, fibers, films, and these can have different adverse impacts of these emerging contaminants. And these contaminants, including nanoplastics, may lead to cancer, for example, may lead to cirrhosis in the cells. It can cause endocrine system disruption and problems with nervous system. It can cause oxidative stress and also some problems at reproductive level. Plastic, as I mentioned before, may affect microplastics and small particles as nanoplastic can affect the soil, the physical chemistry of the soil, and then plants, and all the animals at different levels. And obviously, there is an uptake mechanism of these nanoplastics. The toxicity may be then due to the
Hello.
Plastic that we are using is bisphenol A that has been used in many consumer products, including plastics and food packaging. It is an EDS, what means that it is an endocrine disruptor. And these are the routes of exposure for bisphenol A derivative. I mean, oral exposure, dermal exposure, and inhalation. For example, considering the bisphenol A used in many consumer products, including food containers, for example, it is reported that this can cause prostate and breast cancer, brain and behavioral problems, heart problems, obesity, diabetes, and some other pathologies. So bisphenol A has been used in many consumer products, and the three ways of exposure to the human body. For example, as I already mentioned, for overweight and obesity may occur disorders in the reproductive tract, like ovarian and uterine hypertrophy, premature ovarian failure, reduced semen quality was reported. And in addition, there are other disorders like neurological and hormone-dependent tumors. So, using this picture, we can underline that nanoplastics, such as bisphenol A, used for different purposes, may affect different organs and systems in the human body. Through diet, for example, we can uptake these nanoparticles because they are present in containers and food containers. And there is also parental and childhood exposure that affect different organs and systems, inducing, for example, heart problems, hypertension, and others. But there is ways to determine the effect of BPA metabolite in sample. So here there are some techniques that allows us to identify these elements, these pollutants in food samples. For example, it is also reported that maternal exposure to these endocrine disrupting compounds in the early stage of development and its effect on pregnancy, complication, and birth defect. So there is a high risk for pregnancy and fetal disorders such as miscarriage, polycystic ovary syndrome, uterine hemorrhage, and other adverse effects like preterm birth. So these pollutants affect the embryonic stage, the placental, there is a placental response, and then altered hormone, metabolic, and epigenetic response as well. Several reports point the relevance of some methods to identify these compounds in the breast milk and also in other matrices, such as urine or nails or hair samples. You can see different research studies showing some methods, proper methods to identify and quantify the levels of for example, bisphenol A in urine sample. Our group, we conducted the first reproductive science and fertility report summit during 2021, and we conclude that most of the pollutants present in the environment, in the water, and different places can affect reproductive function, both male and female. And more recently, we also organized at the Royal Society of Medicine in London a workshop where we underlined the role of lifestyle and pathology of male reproductive organs. We have shown that pollutants such as nanoplastic can affect male fertility and can affect female fertility as well. So, fragmentation of contaminants related to personal protective equipment, they have impact on environmental, animal, and human health. So these are some related contaminants. And polystyrene, nano, and microparticles in different biological models do not present cytotoxic effects in both sizes and do not induce DNA damage. For example, in this work, we can see different sizes of micro and nano particles. And different multi-endpoints were studied to evaluate the toxicity of these nanoparticles in vitro. And there are some concerns related to the embryo and genotoxicity. So intercellular distribution, there is intercellular distribution, low embryo toxicity, and non-genotoxic effects. But there are also safety concerns about nanoparticles in vivo application, since they are these applications are growing. And it is a black box concerning the potential nano-hazard, because nanoscale particles can move into the bloodstream and travel to many organs, including the brain, the kidneys, and other organs as well. There are many ways to test biological impacts of nanoparticles. For example, using in vitro studies, using the zebrafish commonly used for testing, and also these nanoparticles can be tested on animals, different size of animals, and results can elucidate us on their effect. And there are possible concerns because the results show us that there are discrepancies between in vitro and in vivo results. Nanoparticle induced biotoxicity depends on particle features, size, shape, surface chemistry, and charge, and dose, and other factors. So it is important concerning not the nanoplastics but other nanomaterials. It is important to design safer nanoparticles. And because nanoparticles have widely tunable properties, and that's the key to increase the performance and safety at the same time in order to minimize exposure and hazard. And finally, I would like to underline that plastic pollution has multi-potential effects on global health. I mean, human health, animals and plants, animal health as well, and environmental health. So the concept of One Health is very important. In addition, we also aim to get some goals, to obtain some goals from the Sustainable Development Goals of the United Nations, such as the good health and well-being, quality of education, clean water and sanitation, for example, climate action, life below water, life on land, and finally, partnerships for the goal. And as a take-home message and final remark, I would like to focus on health effects and the toxicity of different nanomaterials, including nanoplastics, and their health effects on humans, but also in the environmental, and it is required to make the risk and assessment and management. The methods of protection are also important in order to protect environmental health and human health as well. And finally, I would like to thank again Dr. Pratibha Kumari for your kind invitation to be present in this interesting, important conference. I would like to thank my colleagues from my lab, from reproduction. I'd like to thank my colleagues and collaborators around a little bit around the world, like in Brazil, in India, and in different countries in South Asia. And I'd like to thank all of you for your attention. Thank you so much, and thank you.
So we have come to the conclusion of all the 4 lectures today, and the last one being a very important one given by Professor Mario regarding the nanoplastics, and which is a major health concern globally. So to sum up the session, of all the 4 speakers which we had, we had 1 offline speaker and 3 virtual presentations. To start with, we had Professor Parmar's lecture, which had a detailed insight on the medicinal chemistry, the infection caused by bacterial, fungi, and other microorganisms. He gave an insight on the antimicrobial resistance, which is a cause of global concern, antimicrobial chemotherapy, misuse of antibiotics, details of viral and bacterial infections, and then also he discussed about the antimicrobial testing, leadership and surveillance, the fungal infections, and the most important one, the aspergillosis, which is really a fatal lung disease. Then the second speaker was Dr. Ravindra Vikramasinghe, and he gave an insight on the development of novel therapeutic modalities, about the life science services, monoclonal antibodies, MAPs, ADC, and bioconjugates, which is a blooming area, and highly potent APIs, viral vector, mRNA, and lipid formulation. And he also discussed about the Nobel Prize winners in mRNA vaccine and the emerging therapeutic modalities, which is the future of global well-being. Then the third lecture was by Dr. Anthony Adlagata, which was offline one, and he discussed about how the drug resistance, the viral, it occurs faster than the drug development and the selection of different drugs, the enzyme preparations, the enzyme inhibition studies, and he gave a detailed insight on these factors. The fourth one was a video presentation by Professor Maria De, and she gave an insight on the nanoplastics and its adverse effect on the health, which is really a major concern, and what are the adverse effect of emerging contaminants and its possible solutions. So in all, it was a very informative session, and I thank my colleague, Dr. Shubhra, for her, help in assisting me, and the organizers as well. And so we come to the conclusion for this part. Thank you.
Thank you so much, sir. So I take this opportunity to thank Dr. Shailesh Narayan Sharma and Dr. Shubhra Chaturvedi for accepting our invitation and chairing the session in such a nice manner. Thank you, sir, and thank you, ma'am. And my thanks to all the speakers who joined us today in offline sessions as well as virtually. Thanks to the audience who patiently listened to the talks and interacted with the speakers with some interesting questions. Now I would like Dr. Archana Malhotra ma'am to felicitate our chairperson, Dr. Sharma, please, sir, with a memento and a kit. I would now request Dr. Shubhra Chaturvedi to please come forward and Dr. Archana to felicitate our next chairperson for the session. Thank you, everyone. There's a small announcement that after this session, there's a high tea organized in the committee room for the delegates. And we will reassemble here. I think Archana ma'am will